early on in her career, she was offered a position at the EMBL in Heidelberg, where she spent about 8 years, and then got a very renowned position as a director of the Max Planck Institute, where she is till today. Elena has received several prizes, which out of those I would like to name Louis Jantet Prize, that is almost like a Nobel Prize for medicine, sorry, in 2014. And she is also a member of, she was an elected member of the EMBO, European Molecular Biology Organization. And she spent most of her career researching complexes and proteins and machineries that are somehow related to RNA, which most of you know is the most interesting and exciting molecule. So today we will hear about her studies, and I'm very happy that Elena could come to give the lecture. Thank you.
So first of all, let me start by thanking you. Really, it's a great honor to give this lecture. And also, I had a great day today. This is really a stronghold for nucleic acids and structural biology. And so I had a great day today. And I've been asked actually now to say a few words to the students here, on how I got to be a scientist. And I wish I could say something very interesting, but actually I'm very normal. So I grew up, I never thought to be a scientist like, you have these people who say, I wanted to be a scientist when they are 4-year-old and they do experiments with a little chemist. No, I actually went into chemistry because I wanted to go into industry like my father did. And then I got in love with science and I stayed with this. And actually the passion has grown since. So for me, it has been an acquired passion that I still follow. So what I would like to do today is tell you a little bit of the passion that I have for complexes and molecules. And so, 150 years or so after Mendel was thinking about these invisible factors, of course we know now DNA is a permanent feature of our cells. This is what RNAs and proteins are transient molecules. They are made by complex molecular machineries, and they are degraded also by complex machineries in the cell, and these are some of the main ones in the cell. And what I would like to focus on today is our studies on one such machinery, the RNA degrading exosome, and what I would like to tell you is how it is coupled with the protein synthesis machinery. Now what we are interested in is molecular mechanisms, and so a lot of what we do is biochemistry and structural biology to try and understand the chemistry of these reactions and of these interactions. Now, this slide probably in this audience is redundant because you obviously know from the work of Stepanka Vanakova about this molecule, this complex here is the RNA degrading exosome. This is a machine that is present both in the nucleus and the cytoplasm of all eukaryotes that we know today. In fact, it has relatives in prokaryotes, in archaea and bacteria. And it's involved in all RNA degradation pathways that we essentially that we know today. What it does usually is, is shown as this little Pac-Man that recognizes the free 3' end of an RNA, and it chews it up, one nucleotide at a time, until it gets typically to the end. And this is true for RNA turnover pathways, which are known to regulate RNA levels in the cell. There are lots of RNA quality control pathways in our cells that have to detect defective RNAs and eliminate them before they're made into proteins, into defective proteins. But it's also involved in RNA processing pathways in the maturation of precursor RNAs. And there are a kind of conceptual difference between these two pathways, RNA turnover and quality control, and RNA processing, because in these two pathways, the exosome takes over from the 3' end and just chews up until the end. In RNA processing pathways, as I will show you at the end, it's involved in the maturation. So it starts degrading at a specific point in the maturation of the precursor, it degrades, and then it stops. And when it stops, it always stops at the same time, at the same place. And the question is, of course, how can the same complex know what to degrade, and when to stop? And when it has to stop, how does it know how to stop at the same place? So let me take a step back and tell you how it was discovered. It was discovered by David Tolleray at the end of the 90s in C. elegans. In Cerevisiae, and at the time they were actually following a ribosomal RNA processing defect, and they were using purifications from these yeast strains, mutant strains, and actually I think it was one of the first complexes that it was fingerprinted and mass spec by Matthias Mann. And what they found, it was a complex that at the time contained all exoribonucleases. And as you see, this actually has changed quite dramatically, this view, because at the time it was thought this was a core complex of 10 proteins, all proteins as exoribonucleases. And so, of course, the question is, well, why do you need 10 different exoribonucleases? Do you choose one for a different substrate? How do you prevent all this, huge complex of all these exoribonucleases from digesting the entire cell? Now, at the time already, it was clear that this is a conserved complex, and in fact, as similarities to proteins that were purified from the serum of patients suffering from scleroderma pigmentosa. So what is this core complex? So let me start with the complex, with the core. So the core, as I said, is 10 proteins. And actually a big surprise, and largely came from structural studies, in fact, is that 9 of these proteins in yeast and human are actually catalytically inert. They have no catalytic activity, although they have similarities to RNA degrading enzymes in bacteria. And in fact, in yeast and human, there is a single ribonuclease that is attached to the complex. I say yeast and human because Arabidopsis, and I know there are Arabidopsis people here, is different. The Arabidopsis exosome looks more like an archaeal exosome in that it still has retained some ancient RNA degradation sites here, although the function is still unclear. So these 10 subunits, they are all 10 different proteins, and there are various diseases that have been associated, and mutants with exosome subunits, and somehow it has something to do with a motor neuron disease. These are mutations that have been found in patients. It's a disease, this pontocerebellar hypoplasia, that is found in children. And as you see, they map to different components of the complex. So this is the human complex. Now, for a lot of our work, we worked on the yeast complex as a model. We think, and we have data now, I'm not going to show you today, that the human complex for a lot of the basic molecular mechanisms that we have discovered for yeast will look similar. But for today, I'm mainly going to talk about the yeast complex. So here again are these 10 subunits. Again, there is a ring of these 6 gray proteins. These are RNase pH-like proteins. They have no active sites. There is another ring with these 3 proteins in this warm color, which have folds that are typically found in RNA binding proteins, and then the RNA subunit here. All subunits are essential in vivo in yeast. So several years ago, now 5 years ago, using X-ray crystallography, we determined the crystal structure of this complex. In fact, it's not a 10 subunit complex. We had to add, counterintuitively, we had to add an 11 subunit to solve the crystal structure and stabilize it. And what you see here is that, now I see I'm using too many buttons here. And what you see here is, let me just see, yeah, it's a barrel of these gray proteins. You see the ribonuclease in pink at the bottom. The RNA is in green. It's coming down in the central channel of the barrel and going into the active site. So, well, what did we learn about this? Now, it's a lot of atoms, so let me go to a kind of more schematic view. So there is a barrel-like structure of 9 subunits, which is shown here, and there is a central cavity with a narrow entry pore, which is large enough to basically get single-stranded RNA inside. It's not big enough to get double-stranded. And the channel goes in and goes on the side, actually, to get to the ribonuclease active site. So the interactions with the RNA are mainly up at the top and at the bottom. They are sequence unspecific. They are done with they are engaging the phosphate backbone of the nucleic acid. Now, there are strong binding sites at the top and at the bottom. There are weak binding sites in the middle. And this, we think it has something probably to do with the processivity of this complex. And this is true for every molecular machine that has to hold on and has to do a reaction. So it has to hold, it has to cleave a nucleotide. The nucleotide has to be expelled from the complex, and the nucleic acid has to move. And so you have to hold and move. And it's similar, it reminds me actually of the nuclear pore complex, where you have, again, strong interactions at the top and at the bottom, but very weak interactions in the middle to let the molecule go through. Now, the active site is shielded, so there are topological restraints, and as I said, only unfolded RNA can go through. And in fact, all these conceptually are reminiscent of the proteasome. In fact, I think the word was coined by Roy Parker many years ago, when we didn't know about the structure of this complex, that this would be a kind of proteasome for RNA. And this is, of course, only conceptual in this aspects here, because, of course, the folds involved, the chemistry involved, and so on is very different, of course, for protein degradation. But conceptually, these are similarities, which, of course, also extends, and you will see later, also in how the nucleic acid, the substrate, is unwound and unfolded at the top of the complex. Now, if we look at the structure and we see, well, how long does it take for an RNA to go from the top to the bottom? It's about 28 to 30 nucleotide. And this actually is something we knew before, and this is, we had done biochemistry before doing the structure, and from the biochemistry we knew these are RNA's protection assay. We knew that the footprint of a substrate into the exosome is about 30 nucleotides, and these are, kind of, proteolysis for RNA. We're starting with a long, with a 60-mer RNA. We incubate with the exosome. Of course, we need an active site mutant, because this otherwise would chew up the RNA. We incubate it, and then we treat with RNases, and then we see what's protected by the core complex, and this is about 30 nucleotides. And already at the time, we knew we were up to something, and the reason we knew there was something interesting about this path is that there were old data from Scott Butler's lab in vivo in yeast, and Scott was looking again at ribosomal RNA biogenesis, and what he noticed is that knockdown on one of these subunits here, this is a nuclear subunit, which I will tell you about in a few slides, it gave rise, this is a 5.8S ribosomal RNA, is a ribosomal RNA of the larger ribosomal subunit, and when you do not have this subunit, it basically gave rise of this to a precursor, which is 30 nucleotide longer than the mature form. And so already at that time, in 2009, it kind of smelled, we are up to something. So this is a nuclear path of this ribosomal RNA processing is a nuclear function of the exosome. And so I'm going to now move on and tell you where we are to try and understand how the yeast exosome, the core complex, interacts with the nuclear cofactors. So this is the core complex I showed you before. In yeast is present both in the nucleus and the cytoplasm. But what differs is actually the cofactors that it binds to, and these are compartmentalized in a different manner. So there are different cofactors in the nucleus and different cofactors in the cytoplasm. So in the nucleus, in yeast, the main four, and these are conserved from yeast to human, are these four proteins. And again, we have this violet protein and this cyan protein have no catalytic activity, but the red one is another ribonuclease, 3' 5' exoribonuclease, and in this case it's a helicase which unwinds RNAs in the 3' to 5' direction. Now these two RNases are different, RP6 and RP44. This is processive and this is distributive. And the difference for the students is that the processive activity will hold on the substrate and will continue rounds of reactions without releasing the substrate. The distributive binds, makes the reaction, releases the substrate, binds again, does another round of reaction, another cleavage, releases a substrate. And the way we understand it at the structural level is that this correlates to the features of the active site. In this case, this is a very, there is a long tunnel to go into the active site. The substrate is trapped. In this other case, I'm probably not going to go into detail later, as even if you look, you'll see the structure. The active site is very shallow, and it's on the top of the surface, and so things just come in and go. They don't get trapped. This is at least how we naively try to think about it at the structural level. So how do these cofactors go together? Now, at the beginning, I should say we were thinking in this kind of term, but now if you do a pull down from, we are pulling the endogenous complex from yeast. We have a twin strap on RP6, and what happens is that we pull essentially this complex over here. So we pull the core, we pull the ribonuclease, and this goes with its, this is a factor that always binds to it. It's constitutively bound to RP6, we think, and it binds the MPP6 factor. But we, at least in this condition, in fact for us, we will never be able to pull down the MTR4, the helicase. Now if we reconstitute in vitro, and now this is all with purified proteins, we can add MTR4, and I'm not going to show this is published now quite a while ago, and we get a single peak in size exclusion chromatography that corresponds to the 14 subunit complex. So MTR4 can bind, but if we pull the endogenous complex in the conditions where we pull, it doesn't bind. And the way we're thinking about it is that perhaps there is something there where in vivo maybe MTR4 has to be loaded onto the RNA to get targeted with a, to bind efficiently to this complex. Now, because we have this reconstitution, and that should say actually that the first person to, we were working at the time 10 years ago on the archaeal, the first person to reconstitute this core complex here, in fact, is Chris Lima, at the Rockefeller, at Sloan Kettering University. And this, the ability to reconstitute biochemically with purified components, of course, gives us a lot of flexibility in the type of assays that we can do. So in this case, there is an active site here, there is an active site here, there is an active site here, and there is also an active site that we think of, I will not talk about it very much, we think of it as a minor active site also on this side of the molecule. And of course, we can now play games and we can kill one active site at a time, or 3 at a time, 2 at a time, and so on and so forth. And we can study how in vitro this behaves. So we can look, for example, at the pattern of this, the RNase protection as the footprint. This is part of a larger gel. I just spliced the lanes in between because they were not relevant for the talk here. And so what I showed you here, if you have the 10 subunit complex, we are around 30 nucleotide footprint. We add the other 3 subunits that are bound stably endogenously, MPP6, RP6, 47, still 30 nucleotide. Now we add MTR4, we add the nucleotide, and now it shifts much larger. The footprint is longer. And so and I'm not going to show you the data, but for MTR4 to be added and to give this footprint, you need the other 3 subunits as well. So you need all the exo13. And this what told us at the time, and what this seemed to be happening, is that perhaps the RNA goes through the RNA helicase to go down into the exoribonuclease. And so this is the way we have been thinking about it. This is the RNA, and this is just a scheme. And this actually is a scheme that, it's based on crystal structures, that this is, can be loaded perhaps on an RNA and then targeted to the RNA, to the core complex. So what does this look like? So what I'm going to do to take you through is, we'll start with this one, which chronologically is actually the last one we did, but it's one of the last ones. So, with how the core binds MPP6, I will go on how the core then, incorporates this one, and then eventually how we think it binds the helicase. So let me start with this one. How does the core binds MPP6? And this is now the 6 RNase pH-like subunit. Again, these are the warm colors. These are the RNA binding folds. MPP6 is really a silly protein. Okay, it's 180 residues. I don't know, it's 30% or 40% lysines and arginines. It's horrible to work with. And there is a stretch in between that we identified biochemically that binds the top of the exosome complex over here. And here now it's one of the interaction. Again, this is the yeast complex, but this is absolutely conserved. And this is actually an interesting location because this tryptophan here is actually the residues that has been mutated in one of these patients, and actually one of the most severe cases of this pontocerebellar disease in this kind of patients. And what this seems to suggest is it's actually a tryptophan to arginine mutation in this patient. And you might imagine that if you have an arginine here and you have another arginine here, you will weaken the binding of MPP6. And what this seems to suggest is that perhaps there is something with the recruitment of the nuclear factors, cofactors of the exosome, that have something to do with this disease. And I should say the laboratory of Anita Corbett have mimicked this mutation in yeast in vivo, and indeed found that, again, there is something to do, and also Ambro van Hoof, there is something to do with the nuclear function of the exosome. How exactly the nuclear function of the exosome are connected with the disease is, however, still unclear. So this is MPP6. How do the other subunits bind? This is another crystal structure we published a while ago, and here now are the two subunits, RP6 and RP47. Now RP6, again, is one of these horrible multi-domain proteins. And so it has a C-terminal domain, which is you see here. And this is what binds with high affinity to the core complex. It binds on the opposite side of MPP6. So the binding of these proteins can occur simultaneously, obviously. So it binds here on this yellow subunit very tightly. Then there is the ribonuclease domain, which has this bilobal structure, which sits at the top of the core complex here on this orange subunit over here. And then it has an N-terminus, which are these red helices over here. And this forms a very tight interaction with RP47. And this is how it's really like, it's helices going like this. And this is probably why, these two proteins always go together in vivo. So this, you have it up here, it's positioned at the very top. And this is actually, I'm going to play Lego now, and what I'm going to do is to superpose crystal structure of different parts that we have done at different points. So here I'm superposing it with another crystal structure we had done. And what it does, it binds this silly little thing here. This is the N-terminus of MTR4, which binds in this cavity at the top. And this is somehow connected with a linker to the helicase domain. And this is now, is not Lego anymore. Now this is wishful thinking, because at the time that we were looking at this, we of course had no idea what this would look like and how this domain, this whole thing would be positioned on top of the complex. So what is this helicase? Well, it has a classical helicase core. This is in black is the RNA is coming in. This is the 5' end. This is the 3' end. This is where the RNA gets unwound in this direction towards the 3' end. And then it has this arch domain, which is a characteristic feature of exosome helicases, with this domain over here. This is a small globular domain called the KOW domain, which actually has similarity to domain found in ribosomal proteins. So this thing comes in. This is the 5' end, the 3' end, and this is, and so conceptually the way we have been thinking is that this thing somehow will probably sit onto the complex in such a way that the RNA comes from here, it's unwound from the 3' end, and then the 3' end can go into the channel and into the active site. And of course the question is how does it work together? How does this thing work together with the nuclear exosome? And also, you know, all the work we have done so far, all the work we have done, I have a tendency to run. All the work we have done so far is with mimic, substrates mimic, you know, how, the other question is how does it recognize its physiological substrates? And although in the first slide I show you this very nice line of the RNA with the 3' end and so on, of course, the RNAs are not alone in the cell. They're covered, you know, they're part of ribonucleoprotein complexes. They're covered with proteins, and in fact, proteins have something to do with the recruitment, and we know this already because there was a paper from Ed Hurt lab a couple of years ago showing that for the functions of the nuclear exosome in the processing of ribosomal RNA, they identify a ribosomal processing biogenesis factor, NOB53, which binds to this region, to this KOW, to this domain, which is a specialized domain in this helicase. So how does it work? How does it interact? How does it recruit the factor? Now, for me, it's you know, we have been thinking the opposite. You know, we approached it in the opposite way. So usually with, you know, biochemical reconstitution, with crystallography, and with standard, you know, with the classical techniques, you know, the way we are thinking is, you know, what is the smallest possible complex interaction that will answer the question that I have in mind. Now, of course, there has been this huge revolution in structural biology with cryo-EM. Now, with cryo-EM, it's the other way around. So the way you have to think is, what is the largest thing that I can get hold of that, you know, that makes it easier to see? And in the case of the nuclear exosome, that's fair, not easy, but the largest thing is actually the pre-60S ribosome, which is the largest, probably a more complicated substrate of this complex. And this is a paper that's coming out soon. So let me go back at the pathway. So what does the exosome do? And this is work from many years and from many different labs, and David Tollervey and so on this complex. So now I don't go backwards here. This is very complicated. And for me, I should say, you know, I never thought I would work on ribosomes. I'm not particularly interested on ribosomes. It just, for me, it's a perfect substrate of the exosome. And so we start with a precursor, which gets cleaved by an endonuclease, which actually has also been identified recently by Ed Hurt lab. And it leaves around 140 nucleotide more at the 3' end as compared to the mature, and this is where the mature cytoplasmic mRNA, 3' end of the 5.8S RNA will be. Then we know the exosome can get hold of it. And what we know from genetics and biochemistry, biochemical studies over the years, is that the processive subunit, the pink protein, goes on first. It degrades it. It goes down to 30 nucleotides. At 30 nucleotides, something has to happen because it hands over to the red protein, to the distributive ribonucleases. And now this processes it, not to the very end. It goes to 6 to 8 nucleotides. And at this point, this is exported to the cytoplasm where the last processing reaction takes place. And this is actually up here. It's exactly, you know, the northern blot I showed you in the first few slides. Now, of course, you know, in the past, we were thinking about you know, this pathway in terms of nucleotide level. But in fact, as I told you, this is, you know, we think about now in terms of ribonucleoprotein particles. And so these are all going to be covered up with protein. And there is something here, you know, where these complexes get remodeled. Not only the RNA gets degraded, but also the protein composition of these complexes gets remodeled as the RNA gets unfolded and degraded. Now, a lot of this has you know, there's been a lot of work with mass spec, you know, where people have been purifying these substrates and mass specking this substrate to understand what is the protein composition. As it turns out, actually, I think cryo-EM is the best method to understand what's there, because you actually see what's there, and as you will see. And so we saw actually something that we didn't expect. So how did we go about it? So we teamed up with the lab of Ed Hurt, and so what they were doing, and what we have is to try and get this intermediate. So they were purifying this NOP53 pre-ribosomes from yeast, so they have their pre-ribosomes, and then we have all our nuclear exosome with all these active site mutations in one place, the other place, 3 places, and so on. And then we mix them, and then we see what happens. Yeah, and we see both at the this together with Lisa. We could see biochemically, and also Lisa was checking here with either on Comasi bands, but also on northern. And what we ended up doing is to go here. So what we wanted to stall is something around here, and it's easy to do because we need to have the pink subunit active and we need to have the red subunit inactive, and we know how to do this. And so by reconstituting the nuclear exosome, a recombinant 14 subunit nuclear exosome with an active 44 and then inactive RRP6, we can start with an endogenous pre-60S substrate which has been enriched exactly at this maturation state by purification with this factor, incubate it and see what happens. And so this is what we did, and these are what we put then in the EM, and these are now the reference free to the class averages from the EM. And what you see here, the big mass here, is the pre-60S ribosome, but you already see that on the side there is something that is more flexibly bound that is present on the side. And this is now work by Sebastian Falk and Jan Schuller, which has been a very nice collaboration with Lisa and Ed from Heidelberg. So after quite some work, we have collected data on the best microscope that we finally have in-house. It's a Titan Krios with a very good camera. And what we find is a structure. This is a map of the pre-60S bound to the nuclear exosome. So this is the ribosome precursor here. This you'll see later is the nuclear exosome over here. Now, we are overall at 4.1 angstrom resolution. Now, in cryo-EM, this doesn't mean very much because the resolution varies. And in fact, if you look now at the ribosome, this is like a rock, kind of thing. So this is overall 3.9. It's even higher in the center. The nuclear exosome overall is 4.6 angstrom, is better defined here than here. And I say, so for this, of course, we wouldn't be able at this resolution to build a protein structure de novo, but because over the years we have solved crystal structures of various bits and pieces, 4.6 angstrom is enough, as you will see, that we can dock them and see how they fit. And this is how they fit. So this is the pre-60S, and it's a poor choice of color, I can see that. So in green, what you see here are biogenesis factors. These are factors that will go away at one stage or another during the biogenesis pathway. This kind of pinkish color over here is what stays. Now in black is the 5.8S. It wobbles around actually and then goes into the RNA, into the exosome. Now blue is the helicase. Here is the core of the exosome. This is the ribonuclease. The 44 is the least ordered part of the molecule. And here is the ribonuclease over here. And this is now, without the mess of the map. So I'm just going to show you one aspect of this, and that's the aspect that I find most interesting, and that is how the nuclear exosome interacts with the pre-60S. So this also gives you a feeling of what is 4.6, what this resolution looks like. And so what happens is that actually this is the helicase, which is sitting here. Below you see here the exosome core. This is now the MPP6. we actually see an additional feature that we knew also from work of Chris Lima's lab. We knew this part of the molecule somehow had to interact with the MTR4, and indeed we see with C density here. And what you see is that there is a kind of bidentity interaction between the helicase core over here and also here the KOW domain, which interact with the 25S at different time. And actually what happens is that the arch domain, which we saw kind of hunched down in the crystal structure, opens up to bridges the two sites. And we were very pleased when we saw this, because actually we had done NMR with Michael Sattler, we plurale majestatis, I'm Michael Sattler. We collaborated with Michael Sattler to do NMR of how this domain here would interact with the double-stranded RNA. And what Michael found that actually there were chemical shifts exactly at the site where we think it interacts with the 25S. This was also satisfying because we had done in the past, when we saw previous structures, we have done mutations to understand, and there was one mutant of this of RP6 that is here at the back that we could never understand. It's a phenylalanine that points out into solvent, and this gives the strongest phenotype in 5.8S processing. And actually what happens is now we find it here. It's here at the hinge interacting with this arch domain. And also, as I said, we see this N-terminus of MPP6 that we know it's important from Chris Lima's lab. But actually what's even more striking is that there has been a big rearrangement both of the pre-60S and of the ribosome. So there is an EM structure from another group that came out a couple of years ago of a pre-60S at the previous stage. And what happens in that structure, I'm not going to show you here, but you would see green all around here. Okay, so this RNA, it's actually the black here RNA, so it's going to be all cobbled up. This is going to be all biogenesis factor, and of all this biogenesis factor, we only see one, NOP7. It's a single factor that we see now remains bound. So the exosome must have come and remodeled not only the RNA, but the proteins bound to it. And there has been a big change also in the exosome, in the complexes. What I showed you before is the RRP44 was sitting here. Actually, it sits on exactly the same, in the previous crystal structure, it sits exactly on this surface. And now the binding of MTR4 exactly on the same surface has displaced RP6 out, the ribonuclease domain out. So let me just go here, and if I can go to this, gives you a kind of flavor of the structure, so that we start here. I think it's going to now go into these molecules and how we fit in. Now it's going to rotate, and now we're going to look down. As you see, this is the 5.8S in black, and it's coming down. It's coming down from the pre-60S ribosome, and it goes down into the helicase, which sits in this funny way. It's kind of edge-on onto the exosome, and indeed threads the RNA down into the core complex. We see the RNA coming in. The RNA then gets into here, and then we see disorder, the central of the channel, the RNA is flexible over here, and now it's going to go, so it's going to go down here, it's going to go down into the exosome, and now we're going to zoom out. Now the density here is the 5.8S RNA, and you see it inside here, coming up from here, going down into the complex. So this is where we are now, and we see more and more features of this complex. And so what we think is, and this particular conformation is actually relevant, probably not just for these substrates. We think it's probably relevant for different substrates, and in fact we think it's relevant also from preliminary data we have also for the human complex. And so the way we are thinking about it is that at resting state, the exosome, the 13 subunit exosome, will look with a 6 on top, on top of the complex, and somehow the binding, we think the crucial factor is really MTR4, which recognizes probably in this case the pre-60S RMP, but probably it will recognize different RMPs, and in doing so channel the RNA into the exosome, displacing RP6 out into solvent. the ribonuclease domain over there. And so this, brings me to almost the end. We started many years ago. I think when I started 10 years ago, we were looking at the archaeal exosome. There were crystal structure of all the these major machines that are involved in the synthesis and degradation of proteins and RNAs. And we now know how the exosome is structured. These are now in scale. Just to give you a flavor, actually, this is only the small subunit of the ribosome. In fact, we have now gone to try to understand how these complexes are regulated. So us and Chris Lima's lab, we work on the nuclear and cytoplasmic cofactors, but of course there is a lot of work from Patrick Kramer, Venki Ramakrishnan, Wolfgang Baumeister, and so on and so forth, to try and understand how these complexes are regulated, and the regulators are actually, in some cases, as big and complicated as the complexes. And I hope I gave you a sense that actually, there is more excitement to come, because now, the biochemistry has developed to such a stage that we can get bigger and bigger machines. And now we have this wonderful tool of the cryo-EM that is the perfect, has just come at the perfect time to try and understand this machinery. And I think the excitement for the future is to try and understand how these different machineries work together. And I should say, I told you about the nuclear exosome, but we know that the cofactors of the exosome in the cytoplasm are also connected to translating ribosome. And so this is what we are trying to do, to try and catch the cytoplasmic exosome together with the cytoplasmic translating ribosomes together. But I think there is more to come. We know the exosome is somewhere there at transcription. So, to try and catch them with different type of complexes. So I think it's exciting times to come. And with this, I'll just finish with the acknowledgement. So this is, the work has really been pushed by two fantastic postdocs in the lab, Sebastian and Jan. Sebastian has done a lot of work on different types of nuclear exosome subunits. He's a fantastic biochemist and crystallographer. And Jan has joined, and he had a lot of EM experience, but no biochemistry. And it's really the synergy between these two. They taught each other a part of technology that they didn't know that really made this available. And these people have worked with me for a long time in different aspects of biochemistry on this complex. This was a collaboration, very nice collaboration with Ed Hurt and also with Michael for the NMR. And we also thank Patrick, actually, who gave us access to his fermenter in Göttingen, because of course we had to get huge amounts of pre-60S to be able to do this work. Our crystallization facility with Jérôme, we have a great TM facility now with Mike Strauss, the synchrotrons, funding, and you for your attention. And we'll take questions. Yeah. Yes.
I think it's really amazing that we need the structural studies to understand the function of the proteins.
Yes, but we need to know the function before we start to understand how is the mechanism. In fact, I sometimes joke, it seems every X years, everything I touch ends. And the reason is that we can come in when the biochemistry and the genetics are done and the function is known, and it's actually the end thing. So it's somehow we close the subject, it's like, and then you have to start again.
It's a bit unfair.
So then we rely on you to get new things.
So I would like to award you with a MENDEL.
Thank you.
For the lecture. Thank you very much.
Thank you. Thank you. Thank you.
And
thank you. Wow. Very nice.
Please, do you have any questions?
Yeah.
Thank you. So it's a beautiful cryo-EM structure. I'm wondering, what is your interpretation on what stops the exosome from ripping the RNA from ribosome?
Yes, thank you. I forgot. Thank you. Yes, it's nothing very clever. It hits a wall. The helicase normally, when we think about it now, it can go through pretty difficult substrates. yeah, and pretty integrated structures. But now here, essentially it stops because it just hits the bulk of the face of the ribosome. It cannot go through the ribosome. So what we think it's happening, and I haven't gone through this, what we think it's happening is that here what we have done is actually we have captured a situation where the exosome has come in, has gone down to 30 nucleotides, and then it releases it. Now it cannot go to the next step because 6 is mutated, the red subunit is mutated, and so it's probably recaptured in the channel over here. So essentially, you know, what we think is happening is that this is, you know, it cannot do anything else. You know, the ribosome is going to push it down, and what we think is that the arch domain actually helps to flip it open, and once it's open, then it will be accessible to RRP6 in a wild-type case, in a wild-type situation. So essentially, it's just too big a substrates for the, what we think, for the RNAs to go through. And the other thing that perhaps is worth mentioning is the, you know, I told you that the exosome finally lets go at 6 to 8 nucleotide. It cannot go to the end. So how is that? And what we think is that essentially this is why it stops at 6 to 8. It's because the helicase is still bound. So the exosome, we think the RRP6 will, you know, can go to this point, and this is when then the exosome dissociates because the RP6 will not be able to go through the RNA over here. And this in turn suggests that then there is the next step where there is a TPases, or, you know, there are other AA plus a TPases that will have to come in and remove the blue subunit and the green subunit over here. And we think there must be a next step, which now at Hurt Lab is looking at.
Elena, beautiful cross-structural work. I was wondering whether you looked at the dynamics of the MTR4 helicase and possibly how it could be regulated. And the second question would be, what about the role of the distributive nuclease, whether that's maybe not releasing the complex? Yeah, so we haven't, you know, we try to look at the dynamic, but at the time we tried to collaborate with the late Klaus Schulten, who does molecular dynamic simulation, but, you know, we were tackling only this part here. It turns out to be, you know, it's too big to try and see. The only thing we can say on the dynamics is by comparing, well, that I can say on the dynamics is that we can compare the different crystal structures, and what we see is that there must be a conformational change of this part of the molecule upon binding the substrate. But, you know, on what time scale this changes, I don't know. And the second thing is the function of the ribonuclease. The ribonuclease will go from 30 nucleotides to 6 nucleotides. And so what we think is happening is that once, you know, this RNA will have to be released, you know, at 30 nucleotides, we think by flipping over. And at this point, what we think is happening is that the nuclease then, you know, comes back to the top of the complex. And here is when it can trim it. But it can only trim it up to here. So it will degrade, but only up to here. And the other interesting thing, I haven't touched it, is that actually the, you know, why does this go on first? And in fact, there is a, you know, when there is the endocleavage on the 7S precursor, there is a cyclic nucleotide at the 3' end of the RNA. And what turns out is that this RRP6 cannot deal with it, but RRP44 can. And so probably this is a substrate that RRP6 is blind to, and it goes down into the RP44. So this is how we are thinking. But, you know, a lot of it's extrapolation from static snapshots that we see.
Perhaps I missed it, but what determines substrate specificity of the exosome structurally?
So there is, how to put it? So as I said, there is no, so all the interactions here are made with the backbone, so they are not sequence specific. However, there must be, you know, there may well be sequence specificity in terms of structure, in that different RNA sequences may give rise to different RNA structures, and that give a different ability to the RNA complex to degrade one RNA or the other. In this case, in this particular case, the specificity is provided by MTR4, in that it's MTR4 that recognizes both proteins and RNA features on these particular substrates. Now, this is one substrate. How it does it on other substrates is unknown, but this is a crucial component, and in fact, it's the component that interacts with the, forms the next complex, which Stepan has been working on, the PAX complex, and so on and so forth. So this is, we think of it as the crucial element that will bring the specificity to the complex. Yeah, exactly. No, yeah, no. I think there may be something to do with, different abilities of different RNA structures to be degraded, so in that the complex may be faster or slower with one or another molecule. But in this case, the specificity is both the proteins and RNA components of the RNP. It's actually, no, I don't think it's, well, maybe, this is not what the complex will look like in the first stage. This is the end stage, because here, there is a whole, this is essentially the pre-ribosome that has been processed. So the ITS2, as I said, is going to look wobbled up here, covered by ribosomal biogenesis factors. And in fact, MTR4 is brought in by NOP53. Now, at this stage, I should say, NOP53 is gone. So even if we pooled with NOP53, once we process it, we are at a different stage. So I don't know what the complex would look like at the docking stage. It's well possible that it's different. Some of the contacts can be maintained, but others will be accessible. Here, the contacts of the helicase with the side of the molecule will only be accessible once it has been, when it has been changed. So it's a combination.
So maybe I have a question. You started with the link of the mutants in the exosome subunit to the autoimmune diseases. So is there any way to speculate what?
I think it's
since you know now large structures.
Yeah. So I think it's a it's a fascinating thing, and I think it's it's going to come more and more on how it can on how it's connected. So far, it's actually. A lot of this has been connected, at least so far, with the cytoplasmic exosome, in that subunits of the SCI complex, two subunits of the SCI complex mutations are associated with a completely different disease. It's not neurological, it's actually, I cannot even pronounce it, something. It's something to do, again, with it's something to do with the immune response, but it's very early days. I think the field actually are merging now, both on that side and on the RNA degradation side on this, but it's too early days to see what's happening.
Because it must be something more specialized, since it doesn't kill the whole organism, but it's the autoimmune.
Yeah.
Mm-hmm.
Yeah.
Someone has a question?
No.
I was just wondering what first latches it onto the ribosome. Is that place where it binds double-stranded RNA, is that a general double-stranded RNA binding domain?
Yes, that's a general double-stranded RNA binding domain, and here actually there is the binding site for NOP53, which we also done the crystal structure of, and NOP53 NOP53 is in the previous precursor, has a bit that just goes inside the ribosome and then has a bit that is outside, and that's the part that binds the KOW domain. And we know from the NMR studies that it can bind both in the presence, either by itself or in the presence of double-stranded RNA. So what we think is that perhaps at the docking stage, you can have both binding of double-stranded RNA here and binding on NOP53. There is going to be a specific protein-protein interaction that brings it in. But NOP53 then is remodeled in the stage that we are looking at, in the snapshot we look at.
Any other question? If that's not the case, I would like to thank you again for
Thank you, and thank you again for having me here.
Thank you very much all for